goes in than comes out. So that's what we mean by osmotic pressure. We've been talking about the walls of blood vessels. They're really pretty simple. Inside is the endothelium. That's a simple layer, simple squamous, but it's a special epithelium that doesn't cause clotting. Very important. Can you imagine if all the blood vessels clotted? That'd be the end. Then outside of that we have what's called the tunica intima. That's just a little bit of connective tissue behind the endothelium to support it. Outside of that, in the case of arteries, we have a layer of elastic tissue, only it's not a fiber, it's actually a sheet, like a rubber tube. The rubber tube has little holes in it. I'm not sure why, I guess maybe to allow things to pass through the wall. And then we call that the internal elastic lamina or sheet. That suggests that at least occasionally there must be an external one, or we wouldn't call it the internal. And there is in some vessels. Then we have a muscular layer called the tunica media. It could be a mixture of muscle and collagen. And finally we get out to a connective tissue investment of the vessel that kind of makes it tough and strong. And that's called the tunica adventitia. So there's a intima, media, adventitia. There's the aorta. Boy, there's your elastic artery for you, the ultimate elastic artery. The endothelium is way over here on the right. You can't even really see it. It's too small. There's a kind of a thickened intima here. And all of this would be the tunica media out here. And the tunica media has some muscle in it, but an elastic artery, it has lots of elastic laminae and sheets in it. And that allows that aorta to store energy from systole and keep the blood going. If we stain for elastic fibers, there's a special stain that makes them show up. They're showing up in blue, lots of elastic laminae and fibers that are in the aortic wall. This is the adventitia out here, just some connective tissue on the outside of the aorta. If we go up to a high enough magnification, we can see the endothelium lining the aorta. You mainly just see the nuclei, notice that? But you have to trust me, there's a cell going across there. As I mentioned, these cells do not cause blood clotting. There's only one other cell that I know of that, well, two others, that blood can base and not cause clotting. One of the two others, very fortunately, is the surface of the placenta. That would be dreadful, right, if all the blood clotted in the placenta. So the syncytial trophoblast, that layer that coats the placenta, that also does not initiate clotting. These are probably smooth muscle nuclei out here, and then layers of elastic tissue. This is a typical muscular artery here. The endothelium is so thin you can barely see it. You just see a nucleus once in a while. There's a little bit of internal elastic lamina there. And this is mostly smooth muscle. You can always tell smooth muscle because if you look carefully, the nucleus will have a kind of a corkscrew. That's because it's contracted. And by the time this animal or whoever from which this was taken, it could have been an animal or a human, it'll be contracted by the time you get a section and cut it. And then this is adventitia out here. Here's a muscular artery that's going left to right. We're looking at, longitudinal sections running along this way. And we can see those smooth muscle cells that wrap around, sort of barber pole fashion. And these little rings, you see the nucleus is in the middle of each smooth muscle cell. Internal elastic lamina, here are the red blood cells. Adventitia. Here is an elastic artery that has the elastin stained black. And you can see there's an internal elastic membrane and an external elastic membrane. And the reason it looks so zigzaggy like that, it's kind of contracted and kind of folds. Looks like a ripple chip. You ever eat ripple chips? Here is a very small muscular artery. Now you can see the endothelium pretty well there. Here's a couple of nuclei. You can see the red blood cells in here. These are the smooth muscle cells. When you get down to one layer of smooth muscle cells, we call that an arteriole. That's the last vessel before you go into the capillaries where you can start doing some exchange. Another arteriole. This is a true arteriole. One layer of smooth muscle. See the nuclei there and the smooth muscle cells? They're going around Here you can see them wrapping over. See over the top where it's going out of the plane of section. This is a neat picture because it shows what I was trying to explain of the arteries and veins going together wherever they go. This is a whole mount. It's not a section, so we're actually seeing the arteries and veins. And they're running through what we call a mesentery. Remember I mentioned the pushed-in ball? The mesentery of our body, if this is, say, an intestine, it sits in here. The mesentery is where this intestine got pushed into the pushed-in ball, and it's on a kind of a sling here. And up here, it goes to the body wall, like this, okay? And where the two layers come together is called the mesentery, and all the blood vessels going to the intestine have to go down through that mesentery. You can't get in from over here. And so what we're looking at here is a mesentery like this with the blood vessels going through it. And you notice there's a big lumen vessel and a little lumen vessel. What we've done here is we've filled the blood vessels with a dye. So there's a red dye in there. And the big ones are the veins. The little thin red line, that's just the lumen of the artery. So we're seeing the lumen of the vein and the lumen of the artery. Well, the lumen of the vein is much bigger than the lumen of the artery. Does that surprise you that one would be, after all the blood you're sending out has to come back, right? Right. But the blood coming back is under much lower pressure than the blood going out. So if you cut an artery, spurt, 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 right? But if you cut a vein, it just kind of flows slowly and easily. So the blood coming back is going slower than the blood going out. If the blood coming back is going slower, then the plumbing has to be bigger. Or you're not going to be able to get the same amount of blood back as you sent out. And that's just the way it is. Notice when we finally break down the capillaries, all these little red circlets you see out here, those are all capillaries. Notice they're all kind of running by their lonesome. There is no comparable vein to a capillary. But as soon as they go into a vein, we have this double system. If you make a section across right there, you would see this neurovascular bundle that I told you about. Here it is. This is an artery. You can tell by the muscle in its wall. Next door is a vein. If we opened this thing up, it would be much bigger in diameter. It's kind of collapsed there. These things you see running through the middle, they're the valves. The valves are kind of coming at you like this that can open and close, and so you've cut across and they just look like two lines going through there. And then down here, there's our nerve bundle. If you look carefully, you can even see some myelinated nerves in there. So artery, vein, nerve going together, in this case through skeletal muscle. These are skeletal muscle cells cut in cross-section here. Here's a lymphatic over here carrying lymph. Notice you don't see much in the way of blood cells and lymph. You might see the occasional lymphocyte, but you don't see red blood in lymph because that's just bringing tissue fluid back. This is a vein. It has a thin wall, but it's thicker than that lymphatic, and the vein is full of blood. This shows you how thin the wall of a lymphatic is. See it right here, that little thin line? Yeah. Talk about a squamous epithelium. That is squamous. Over here is a vein with blood in it. That's a comparison of a artery and a vein. See how much thicker walled, how much more muscle in the artery than in the vein. Well, I've told you more about blood vessels. Let's just end by showing you those valves in a lymphatic. Which way do you think the lymph goes through? From left to right or from right to left? Just take a look at that and look at that valve there.
Left to right?
Absolutely. You've got it. You know, because in the lymphatic, it's just like the vein. You don't have a heart to get this lymph back. So you have to squeeze it back with the muscles of your own body as you move and walk and what have you. And when it squeezes, the lymph can go to the right, but it can't come backwards because the vein closes like that. And you can see them coming together there. That's a kind of a fortunate shot. And finally, here's a lymphatic capillary in the electron microscope to show you how thin wall. It's even thin in the electron microscope. And of course, the question is, what on earth would hold it open? It should just collapse. If the pressure is less inside the lymphatic than outside, it should collapse. But it's held open by little fibers that are attached to it that pull it open. Well, I've told you everything I know about the heart and the blood vessels and the lymphatic. I don't know if I've left any stones unturned, but if you have any questions, I'll try to answer them, but ask easy ones, please. I hate hard questions. You got any, Matthew?
What would cause an enlarged heart?
That's a good point. We say that a heart should be about the size of your fist, but I had a class where some young ladies that were being homeschooled came into the gross